Hello, and a very warm welcome to today's Cell and Gene Therapy Insights webinar titled Achieving Comprehensive Analysis and Characterization of Extracellular Vesicles. I'm Casey Nevins, an editor at BioInsights, and joining me today is Dr. Alex Shepard, who will explore a method to overcome the challenges of accurately characterizing extracellular vesicles. This novel solution allows for single particle analysis of extracellular vesicles and exosomes in both pure and crude samples. After the presentation, we'll have a live Q&A session. We invite our audience to pose their questions to our speaker using the Ask a Question box at the bottom of your screen, and we'll try to get to it during the session. Now I'd like to introduce our presenter. Dr. Alex Shepard is the market manager for cell therapy at Unchained Labs. She has many years of experience at the bench and as a field application scientist, characterizing exosomes and viruses using various techniques. Alex completed a PhD in molecular biology at Bristol University and held a postdoc position in the tumor microenvironment group at Cardiff University. So without further ado, I will hand over to Alex to kick us off with the presentation.
Hi everyone, I'm Alex Shepard, Market Manager at Unchained Labs, and today I'm going to show you how Leprechaun can help you fully characterize your EV sample and get a complete breakdown of any EV subpopulation, no matter how rare or small. There are several reasons why EVs are particularly tricky to analyze. Firstly, most EV samples are pretty complex, meaning that there are lots of other particles present besides your EVs of interest. For example, lipoproteins, cell debris, and protein aggregates. Therefore, to get meaningful results, you need to be confident that you're analysing only your EVs of interest and not these other contaminating particles. Secondly, most EVs are really small. Most particles of interest will be below 200 nanometers in diameter, meaning that there are few optical techniques that can size down to this level. Thirdly, most EV samples are highly polydispersed and heterogeneous, meaning that bulk analysis techniques such as DLS struggle to provide meaningful data under these circumstances, leaving you needing a single particle technique to be able to get accurate information on your particles. And finally, in a lot of cases, you may be looking for a really rare EV population, meaning that there are very few EVs in your sample with your marker of interest. Taken together, these factors mean that you need a method that is really highly sensitive, highly specific, and can size particles down to the smallest level to be able to find out exactly what is in your EV sample. But even amongst recent single particle techniques, most methods struggle with the unique challenges of analysing EVs. There are several reasons for this. Firstly, the sizing limit of these methods can vary depending on the purity of the sample, struggling to get down below 50 nanometers consistently. There can be limitations to the phenotype capabilities, either due to photobleaching, as in the case of NTA, or even a complete lack of phenotype analysis, as for TRPS. And critically, for all these methods, the quality of the data you get out is only as good as the purity of the sample you put in, limiting their ability to handle complex biofluids or even cell culture samples without significant prior cleanup. Why does this matter? Well, even the best purification method is not 100% efficient, resulting in sample loss, which can bias results, and meaning you need a higher starting sample volume, which is problematic if you're working with low abundant samples. So wouldn't it be great if there was another solution out there which could help overcome some of these hurdles to EV analysis? Well, thankfully there is. Leprechaun is the only platform that can fully characterize EVs from just a few microliters of unpurified sample. In one run, Leprechaun can provide EV concentration, size down to 35 nanometers, and analyze the expression of up to 4 markers per EV, so you can find out everything you need to know about your sample in one go. Even better, it does this on a single particle basis, without the need to purify your sample beforehand. Based on the same technology that underpinned its predecessors, the ExoView R100 and R200, Leprechaun adds improved small particle sizing capabilities to ensure you see even the smallest EVs, along with beefed up, more user-friendly software. So how does it all work? Well, Leprechaun characterizes vectors, including exosomes, on up to 16 samples at a time. You just add a maximum of 25 microliters of your sample to our Luni consumable, which will capture your EVs, and then run the Luni on Leprechaun and sit back and relax while it uses interferometry to individually size each particle and immunofluorescence to analyze the structure. Lunis come in different shapes and sizes depending on your sample type. The human tetraspanin Luni is pre-coated with highly specific antibody spots against free exosome tetraspanin markers, so you can specifically capture exosomes from even the crudest sample. Each Luni features triplicate spots for CD63, CD81 and CD9 to grab hold of your EVs, but if you're working with plasma or mouse samples, there are Lunis specifically tailored to your sample. Each Leprechaun comes with a Luni washer to automate sample prep, capable of washing, staining and drying up to 24 samples at a time. The Luni washer not only significantly reduces hands-on time, but also helps guarantee consistent results and remove user-to-user variability. So once your EVs have been captured on the Luni and stained with up to 3 fluorescent antibodies of your choice, it's time to read them on Leprechaun. Sequential images are taken using the red, green and blue fluorescent channels to confirm which of your chosen markers are present on your EVs. In this example, particles which appear yellow in the fluorescent image express both CD81 and CD63, while green particles express only CD81. Leprechaun sizes up individual EVs using single particle interferometric reflectance imaging, SPIRIS for short. Upgraded optics compared to the XView R200, the previous version of the platform, opens the door to sizing EVs as small as 35 nanometers, meaning you're no longer missing the smallest particles in your EV population. By overlaying the fluorescent channels with the interferometric sizing channel, it is possible to build up a picture of the structure of each EV based on particle size and markers present. This allows Leprechaun to separate particles into distinct populations and provide EV concentration, phenotype, and size for multiple subpopulations in one run. So from just a few microliters of crude sample, you can get all the key info on your EVs in one go, whatever your sample type. No need to spend time concentrating and purifying your sample or worrying about how your purification method is affecting your results. And with hands-off data acquisition, you can avoid spending hours manually loading and running samples. Just sit back and relax and wait for Leprechaun to provide the answers. If you have EVs which already contain the fluorescent tag or molecule, you don't even need to go through the process of staining with antibodies. Leprechaun can detect a range of commonly used fluorophores, including GFP, M-Cherry, and FITC. As an example, analysis of a commercially available GFP EV standards from Sigma shows that Leprechaun can identify GFP positive and GFP negative EVs within a sample, and quantify the percentage of GFP expressing particles. It can also provide separate size profiles for the two populations, which show that GFP negative EVs are smaller than GFP positive EVs, giving you detailed insight into the different EV populations present. Importantly, EV marker analysis is not just limited to the detection of surface markers. Our Cargo add-on lets you look inside your EVs. By gently fixing and perming the particles, you can probe for internal cargo proteins alongside your surface markers. Here we detected the internal protein syntenin, shown in red, alongside CD81 and CD9 on the EV surface. Not sure whether your EV marker of interest is internal or external? Well, by comparing permeabilized and non-permeabilized controls, you can establish the location of any EV-associated protein. If you're aiming to use your EVs as a delivery vehicle and are loading them with a specific protein or small molecule cargo, Leprechaun can measure loading efficiency and quantify the concentration of EVs with the desired cargo. This recent paper from one of our users shows how Leprechaun was used to measure loading of doxorubicin into EVs. On the left-hand images, you can see that CD63 EVs are stained red, and the doxorubicin is intrinsically fluorescent in the Leprechaun green channel. By overlaying the fluorescent channels, CD63 EVs, which are loaded with dox, can be identified as they appear yellow. Size analysis of the EVs showed that doxorubicin loading created EVs that were 10% larger than the empty particles, and loading efficiency was measured at around 20%. So far, we've mainly focused on detection of protein cargo by Leprechaun. However, recent exciting results from Ken Whitworth's lab at Johns Hopkins University show the proof of concept of sequence-specific RNA detection in EVs using the Leprechaun technology. By adding a small molecule fish probe recognizing M-cherry mRNA to the EV sample, they were able to quantify the concentration of EVs containing M-cherry mRNA via ExoView. A Cree mRNA was used as negative control to show specificity of the small molecule fish signal. Therefore, whatever cargo you are looking for, protein or RNA, Leprechaun can identify whether it's EV associated and measure the concentration of particles which contain it. How about if you're looking for a really rare EV population? Well, you may want to customize the EV capture step to help enrich for your EVs of interest, rather than just using the standard tetraspanin capture. Leprechaun Flex kits let you customize the Luni capture step from the comfort of your own lab. All you need to do is incubate an antibody against your marker of interest with our kit-provided linker protein, add it to the Luni, and you have custom EV analysis ready to go. Depending on the protein marker you're interested in, you may not always know if it's EV associated or soluble. Leprechaun can help solve this question. By using the Flex Luni and customizing the capture step with an antibody against the protein of interest, in this instance PD-L1, you can get a measure of the total amount of that protein in your sample. By counterstaining with tetraspanin fluorescent antibodies, you can check whether that protein is bound to EVs. In this example, HEC293 EVs were used as negative control for PD-L1, with the breast cancer cell line HMEC as a positive control. We can see significant PD-L1 positive staining in the HMEC samples, suggesting there is a lot of PD-L1 present in this sample. However, when we add a blue tagged antibody against all three tetraspanins, very few of the PD-L1 particles co-stain for CD9, 81, or 63. This shows that the majority of PD-L1 protein in the HMEC sample is not EV associated. Leprechaun has a lower limit of 5 × 10⁵ particles per mil for EV concentration, making it compatible with samples taken straight from cell culture and a wide variety of biofluids, without the need for prior purification. Biofluid samples will generally require a dilution of 5 to 10-fold in kit-provided buffers, while purified cell culture samples are commonly run at a dilution of 100-fold or greater. With a maximum on-Luni volume of 25 microliters, you'll regularly find yourself using 1 to 2 microliters max of your valuable EV sample. Easy-to-use kits with step-by-step protocols deliver the answers in just a few hours. Each kit contains 8 Lunis, which, as you've seen today, have been specifically designed to immunocapture EVs from your sample. The kit also contains all required buffers, as well as the fluorescent tetraspanin antibodies for counterstaining your particles once bound to the Luni. Our super easy-to-use analysis software gives you all your results in one go and in one place. With automatic background subtraction and data QC, you can be confident that non-specific signal isn't interfering with your data. When you've run all your samples, you can export a report in a couple of clicks to get an easy-to-read overview of exactly what's going on. So come and join the revolution. With over 500 publications in the last 5 years, Leprechaun and its ExoView predecessor are proven to help researchers ID and analyze their EVs in any sample type and for any application. Leprechaun is the unmatched EV characterization tool and the only platform capable of measuring concentration, size, and phenotype on a single particle basis from just a few microliters of purified sample. No other EV analysis technique does as much with as little as Leprechaun. And with that, I'd like to thank you for attending this webinar. If you want to learn more about Leprechaun, don't hesitate to reach out to us at info@unchainedlabs.com. Take a look at our website or ask a question right now.
Thank you so much, Alex, for a great presentation. We are now going to start our Q&A. Our first question is, how many copies of a protein need to be present on the EV surface to be detected by Leprechaun?
So the great answer is only one, So Leprechaun has single epitope sensitivity. We've previously done some DNA origami experiments, which show that Leprechaun is sensitive down to 3 fluorophores in all of the fluorescent channels. So based on the fact that most commercially available antibodies and the ones we include in our kits as well have a fluorescent degree of labeling of at least 3, then it means you only need a single antibody to bound to be able to detect that surface protein. So yeah, 1 epitope is required per particle.
Great, thank you so much. Our next question is, do you see any reduction in sensitivity or reproducibility of the assay in crude samples compared to pure?
No, so the linear range of the assay is unaffected by sample purity. And we see a percentage CV for EV concentration that remains below 10%, whether you're working in crude or pure samples.
Great. Our next question is, can Leprechaun analyze other nanoparticles besides EVs?
Yep, great question. So Leprechaun can analyze most spherical nanoparticles or envelope viruses as long as they're between 35 to 200 nanometers in diameter. Besides that, the only other requirement is that there's a protein epitope on the surface, which you can use to capture the particle. So, for example, tetraspanins on exosomes, or an envelope protein if you're looking at a virus. So in addition to the exosome analysis that we've spoken about today, Leprechaun also has a specific application and set of consumables for lentivirus analysis. So you can measure size, titer, empty-full ratio of your lentivirus in a similar way to what we've spoken about today with exosomes. But if you're working with other viruses or other particles besides lenti or EVs, then our Flex kits let you customize the capture antibody so you can pull down any particle. So, for example, we also previously looked at SARS-CoV-2, rabies, HSV, and other particles.
Thank you so much. That's really insightful. Our next question is, are there any restrictions to the type of EV cargo that can be analyzed?
So we have a cargo add-on to all of our consumable kits, which is really focused on detecting protein cargo inside EVs. But I think we've shown here today that there is precedent that you can adapt this cargo protocol to detect other components such as nucleic acids, including mRNA. And really the key aspect to that appears to be that you need a good fish probe or molecular beacon against your sequence of interest. But if you have that, then you can adapt our standard cargo detection to look at other non-protein cargo. We don't sell fish probes or molecular beacons, but we can show you what previous customers have done and how they've gone about this and how you might be able to adapt that for your particles. So yeah, protein, nucleic acids, other cargo, it is possible with some adaptation of the assay.
That's really helpful. Thank you. Our next question is, which biofluids is Leprechaun able to analyze? Are there any that aren't compatible?
So we have a very specific on Luni capture step via that antibody capture, which is then followed by several subsequent wash steps. And because of that combination of highly specific capture and then multiple wash steps to remove any non-specifically bound particles, Leprechaun should be compatible with any biofluids. Because anything that isn't EVs will be removed during that process. And to date, we've tested almost every biofluid out there: plasma, serum, urine, through to CSF, even tears and sweat. And we haven't yet come across biofluids that Leprechaun can't analyze.
Great, thank you. Our next question is, could you provide more detail onto the permeabilization for the cargo analysis?
Yeah, so we provide a fixative and a permeabilization reagent in our cargo add-on. So what these basically do is the fixative will just fix everything onto the Luni surface to make sure none of those internal protein or internal cargo molecules can escape. And then we use a mild detergent for the permeabilization, so it's not strong enough to break apart the EV entirely, but it basically just punches some small holes into the surface of the EV or the exosome, which then gives access for your fluorescently labeled antibody to get inside and bind to your target cargo.
Thank you so much. Our next question is, how does the number of events/particles you analyze per run compare to other techniques?
Yeah, really interesting point. So each, as you said, the Luni is covered with antibody capture spots, and each of these capture spots combine a maximum of 4,000 particles. So then we have each marker spotted in triplicate on the Luni surface, which means that you can capture up to 12,000 particles per capture marker. So if you can have a maximum of 6 different capture markers on a Luni, which means you can look at around 72,000 particles per sample. How does that compare to other techniques? Well, the number of particles that you might analyze using either NTA or flow cytometry is obviously going to depend on the length of the video or the run that you perform, which can make it a little bit hard to compare results between users if you don't know exactly what acquisition settings they've used. So as an aside, one bonus of Leprechaun is that you avoid this problem by having really standard acquisition settings. So where everyone in your group, in your institute or across users is going to be using the same settings, so you can be sure that it's more comparable from a user-to-user basis. But if you're looking at a typical length run for nanoflow cytometry or NTA, then you're probably looking at around about 2,000 to 3,000 events, say a maximum of 5,000 realistically, which is obviously significantly fewer than the 70,000 or so that you will be analyzing with Leprechaun.
That's really helpful. Thank you. Our next question is, what is the main difference between Leprechaun and ExoView?
So the headline differences I think we highlighted here is really that improvement in the lower sizing limit. So Leprechaun has improved optics, which means it can go down to 35 nanometers in comparison to the 50 nanometer limit for the ExoView R100 and R200. And this makes Leprechaun really the only technology besides super resolution microscopy which can size and count those really small EVs. So then in addition to the improved sizing capabilities, with Leprechaun, we've also redesigned the software, so it's much more user-friendly and quicker and easier to get your results. So if there are any current ExoView users out there today, you no longer have to go through that process of manually setting your fluorescent cutoffs and QCing the individual spots. Leprechaun automates all of this for you.
Great. Our next audience member says, you talked about customizing the capture and detection antibodies to look for your own markers. How many custom markers is it possible to analyze per sample?
Yeah, so we have 3 different fluorescent channels, red, green, and blue. So you can have up to 3 different custom detection antibodies, one in each of those 3 channels. And then on our Flex Luni, you can add up to 2 different custom capture antibodies. So that gives you a total of a maximum of 5 different custom markers that you could be looking at per sample by combining that custom capture with the custom fluorescent staining. So a maximum of 5.
Great. Our next audience member is asking a clarifying question. They say, I noticed that on the fish example, the y-axis was background subtracted. Does that mean I can easily measure my sample as well as the medium and then just subtract the readout as background? And can I do that in the software, or do I export the data to Excel?
Yeah, so on every Luni, we have an isotype control spot. So these are standardly mouse IgG capture antibodies, which complement the isotype of the tetraspanions we're using to capture, or you can customize that if you're using a custom setup. So the software will automatically look at what level of binding you have on that isotype control and subtract that from your positive capture spot. So that is automated. If you are, I think in this example you referenced running the media and then comparing that to kind of a positive sample, kind of a positive and negative sample comparison. You can do that, and in that instance, that would be something where you can then just take the final results and subtract that yourself. So it depends slightly if you're kind of comparing a positive sample to a negative sample, or if you're doing an internal negative control. Because if it's internal negative control within a sample, then that will be done automatically for you in the software. Hopefully that's clear.
That's really helpful. Our next audience member asks, does plasma EV's analysis require an SEC pre-fractionation?
You can do. I think we'd always recommend with plasma EVs, try it unpurified first, because if you can get, you should be able to get good quality positive signal, and if you can, then you're going to be getting more accurate data because you're not introducing any bias from the cleanup step, whether that's SEC or anything else. If you're looking at a very rare EV subpopulation, then sometimes it can be beneficial just to do a cleanup step beforehand. But I would say you always run your sample first, see if you're getting good positive signal, and if you are, you don't need to clean it up. There may be occasional instances where that cleanup step can slightly help you.
Great. Our next question is, what things to keep in mind if we use custom antibody conjugated as detection antibody?
So I think as with any antibody-based technique, the first thing we'd always recommend is some kind of validation of your custom detection antibody. So ideally, either if you've used it on a different technique, we find that antibodies that perform well for flow cytometry generally work very well with Leprechaun, so that can be a good indicator. And then when you test it out, if you can have a positive control, a sample that has your protein of interest in it, then that could be really helpful. As I say, we have an inbuilt negative control on each Luni, so if there is any non-specific binding, or if there's any fluorophore left over that hasn't been removed and is unbound and is sticking to the chip, to the Luni surface, then you will be able to see that, and it might be an indication that you need to optimize that cleanup step a little bit. But also, if you try that and you have any questions over how well your antibody is performing or whether the data looks good, then our technical teams can also help you take a look at that and suggest steps that you may need to take.
Great. Well, thank you so much, Alex, for answering those questions. Unfortunately, that is all we've got time for today. Any questions we didn't get to, we will reply to by email. The webinar will be available on demand tomorrow, so look out for an email from us with the link. All that's left is to thank Alex once more for a great presentation, and thank you to the audience for listening. We hope you'll join us again soon.